Welcome back to the Chapter 4 video. This is going to be a relatively short video, but very important video. We're going to highlight some important lab media from this chapter. There's lots of different media highlighted in this chapter that we will use later in the semester. For right now, I will introduce you to a few different media that will be important for your first exam. We will also use this media in the lab in our first unit. The different culturing media that we use are the general purpose media such as tryptic soy agar. So you use this in lab 1.1 and lab 1.2. There's selective media, there's differential media, and then there's also selective and differential media. So general purpose media like tryptic soy agar is used to grow most lab organisms. And with tryptic soy agar, we're able to determine colony morphology. So here this is a pure culture of Staphylococcus aureus, and what we want to do is use what's referred to as the streak plate method or the quadrant method to actually obtain isolated colonies. So the isolated colonies are groups of bacteria growing from one single cell. So in 24 hours we can have a large colony due to the fast generation times of most of our organisms. So here on the left, this image is a pure culture of Staph aureus. Here on the right, you can see this is a handprint, and on the handprint there's a variety of different colony morphologies growing on this person's hand, or from this person's hand. It takes one cell transferred to this media to form one colony. So this looks disgusting, but if you look down in your hand, you don't actually see this because our skin has properties that limit bacterial growth. So colony morphology is the description of the organism, what it looks like basically. So here for Staph aureus, you see these nice round yellow colonies. If you look at this in person, they might be a little bit shiny. So the morphology is terms, characteristics that you would use to describe the actual bacteria growing as a colony. Selective media inhibits the growth of some bacteria while selecting for the growth of others. So here we have bacterial colonies growing on this media, very much like a tryptic soy agar. This media has a pH of 7.3. Okay, so at neutral pH, we can grow all these bacterial colonies. A lot of bacteria don't like acidic environments, so here with a pH of 5.6, we're actually selecting for fungal colonies. So we're selecting for fungus, we are inhibiting the growth of most of these bacteria because of the acidic environment. Differential media differentiates between different organisms growing on the same plate. So our example is blood agar. We will use blood agar in lab 4, which is tryptic soy agar with 5% sheep's blood. So there's nothing in this media that inhibits the growth of organisms, but the addition of the blood makes it differential. So we use this to differentiate between hemolytic abilities of bacteria. There's alpha hemolysis, which is an incomplete lysis of red blood cells, and what happens is a green zone appears around the colony. And again, you want to look at the isolated colonies to determine hemolytic activity. There's beta hemolytic, which is a complete lysis of red blood cells. So what happens is we get a clear zone around the colony. And then there's gamma hemolytic, so there's actually no lysis and no change to the media. So here we have alpha hemolytic bacteria. You see this green zone form around the colony itself. Beta hemolytic, see this clearing zone. The media appears this yellow color because that's the color of the tryptic soy agar. But the blood is completely cleared, so the bacteria uses the blood as a nutrient source. And then here we have gamma hemolytic, so there's no hemolysis, there's no change around the bacteria. We can combine the properties of selective media and differential media into one. So one of the examples is mannitol salt agar, or MSA. So what happens is we're selecting for salt-tolerant organisms. In mannitol salt agar, there's a high concentration of salt, 7.5%, which inhibits most bacteria from growing. However, some skin organisms, for example, are capable of growing in the high salt concentration. We're differential for mannitol fermentation. So mannitol, found in the media, it's a sugar that not all organisms are capable of using. The organisms that are capable of using mannitol ferment the sugar, meaning they break it down and they produce an acid byproduct. So there's a pH indicator in the media that turns the media yellow when mannitol is fermented due to the acid byproduct. So here is mannitol salt agar, as we'll see it without any bacteria growing on it. So it's kind of this pinkish color. Here we have mannitol salt agar with two different types of bacteria growing on it. The one on the left here is a salt-tolerant organism. So it's growing on the media, but the media you see is not changed in color. The one on the right is a salt-tolerant organism and a mannitol fermenter. You know it's fermenting the mannitol because you see this color change to yellow following the growth and fermentation of the mannitol. So on the left we have non-fermenting but salt-tolerant, and here on the right we have fermenting and salt-tolerant. Eosin methylene blue is another media that we will use, and this is a selective and differential media. It's selective for gram-negative coliforms. So coliforms are fecal organisms. Methylene blue will inhibit most gram-positive organisms. Eosin makes this media differential for two major coliforms, E. coli and Enterobacter. The differential properties comes from the rate of lactose fermentation by the organism. So there's lactose in this media. Some organisms will ferment lactose at a very fast, rapid rate, such as E. coli. In E. coli, you get this green metallic sheen found on the surface of the bacteria. Other organisms like Enterobacter, they ferment the lactose relatively slowly, so we don't get the metallic color, but we do see this reddish, dark black, sometimes centers on the actual colonies. The last portion of this chapter discusses biochemical testing. So every bacterial species produces different enzymes which allow them to produce different metabolic byproducts. So the mannitol salt agar is basically a biochemical test. So since some bacteria can produce enzymes to break down mannitol and others can't, we can use that to differentiate between different types of bacteria. So not all bacteria are created the same. This is an example of another biochemical test that we'll use later in the semester. This is Simmons citrate agar. We're looking to see if the bacteria can break down citrate as a sole carbon source. So the media is normally green to begin with. So this one here is negative, so the bacteria does not use citrate. The one on here turns blue, indicating the bacteria used citrate as a carbon source. So again, differentiating these tests allow us to helps identify unknown bacteria. So later when we do the practice on known labs, you will see this firsthand.